Okay. Hello, everyone. My name is Asif Jamo. I'm from Ethiopian Food and Drug Authority. I'll be presenting on behalf of Group 8 members, Dr. Yalam Zaud, Dr. Zalalam, Nisu, Tapelo, and Tumu, which are our group members. So um, our presentation will be on assessment of evidence for originator product, Remicade versus the biosimilar, that is Felixavi. Presentation outline, we do have case study, which was assigned for our group members. Background on the products and the products that are originator and versus Felixavi. We'll look at assessment of indication, that is, we choose rheumatoid arthritis, and how to handle licensing of Felixavi. And finally, we look the regulatory decision, which we agreed to, just regarding how to accept and or not accept to our agency or regulatory body. So when we come to the case study given to our group was to critically assess doses or evidence for the originator product, that's Remicade versus the biosimilar, that is Felixavi, as it relates to human pharmacokinetics, clinical efficacy, and adverse effects or safety aspect of the product. Under this topic, we do have 3 points to answer. One says identify, the first says identify specific indication, example, rheumatoid arthritis, and quote specific clinical trial data and or evidence from European Public Assessment Report during your assessment. The second one is please reflect on how you would handle the licensing of biosimilar in all indications that we are licensed for originator products, that's Remicade. Finally, it says present summary as to whether you would grant a positive opinion for your agency or not based on the information available to you. Just to start from the background, Remicade, originator and Felixavi biosimilar contains Felixavi, an immune suppressant belong to the class of tumor necrosis factor alpha, that is TNF alpha inhibitors. Infliximab is a chimeric human murine immunoglobulin G monoclonal antibody produced in murine hybridoma cells by recombinant DNA technology. It binds with high affinity. It binds with high affinity to both soluble and transmembrane forms of tumor necrosis factor alpha, but not to lymphotoxin alpha or tumor necrosis factor beta, which is a former name of that of lymphotoxin alpha. Next, we do have for both originator and biosimilars to look at some parameters. Among the parameters, the first one is manufacturer. Manufacturer of the originator, that's Remicade, was Janssen Biologics, that's BV and Stewing, 101 CB Leiden, the Netherlands. And biosimilar one is Felixavi, that is Biogen Netherlands. And also there is another manufacturer for this Felixavi, that is Samsung Biopsys. I think it depends from actually the brand or the biosimilar is Felixavi, but the manufacturer matters actually, which is the two manufacturers, that is Samsung Biopsys and Biogen Netherlands are from Netherlands. When we come to the approval status of approval dates of those both originator product and biosimilar, originator one was approved by EMA on August 13, 1999, and also biosimilar or Flexavi was approved by EMA on May 26, 2016. The indications, approved indications for originator originator products that Remicade were rheumatoid arthritis, Crohn's disease, ankylosing spondylitis, psoriatic arthritis, psoriasis, and also ulcerative colitis, for which it was approved on 21/02/2007. And similarly, for Flexavi, rheumatoid arthritis there, Crohn's disease cells, also ankylosing spondylitis is there, psoriatic arthritis, psoriasis, and also ulcerative colitis. Similar indications are presented. When come to the other parameter, that is clinical trial studies, that is originator Remicade was approved for all of the above mentioned indications, and biosimilar or Flexavi was approved for only rheumatoid arthritis. When we come to the specific questions that need to be answered by our group members, the first one is identification of a specific indication and quotation of a specific clinical data or evidence from EPR during our assessment. Just directly, let us go to that clinical data discussion. Just areas which need to be considered where pharmacokinetics, as the case requires us to do on that one. The other one is clinical efficacy and safety, and also we'll discuss on the reported adverse effects. The identified indication was by our group was rheumatoid arthritis. The reason for choosing this one is there are clinical studies for both products on indications for comparison, that is for biosimilar and also the originator as well. So rheumatoid arthritis clinical study was conducted on both products. That is why we choose rheumatoid arthritis. When come to discussion on pharmacokinetics, clinical comparative human PK study employing sensitive population, doses, and route of administration that allow detection of whether difference in PK exists or not was conducted. PK parameters were Cmax, area under the curve, Tmax, and it was conducted on patient subjects on single dose. When we come to Flexavi, we can look at the same parameters that are Cmax, area under the curve, Tmax. It was conducted on healthy subjects on single dose was used to conduct. When we come to conclusion on that PK study, the PK values for the originator and the biosimilar were comparable with Cmax averaging 126.9775 microgram per ml for the biosimilar and 126.23, or we can say approximately 28 microgram ml for the originator, even though different population was used. When we come to clinical efficacy and safety aspect, clinical comparative safety, including immunogenicity, was conducted. And again, clinical comparative efficacy for potential biosimilar and originator using sensitive clinical endpoints, usually designed as equivalence study, was conducted. On Remicade case, dose response study was done. Efficacy studies done all indications mentioned previously, including ankylosing spondylitis and also Crohn's disease, not only rheumatoid arthritis as case of Flexavi. There was a primary endpoint, which is American College of Rheumatology, that is 20%, ACR 20% response difference within a 15 plus or minus 50% margin in 95% of confidence interval. We can see sample size that 584. The study shows that ACR response at week 30 was 64.1% in SB2, which was SB2 stands for both Flexavi and Renflexis. Renflex, I need to say something on this Renflexis. That was approved in US. Thus Flexavi was approved in. These both are biosimilars to the originator one. That one of Flexavi was approved in EU. That is SB. That is for what SB2 stands for. And also in Flexavi, that is 66%. Secondary endpoints was also conducted on 54 weeks, just like American College of Rheumatology, 50%, 70%, and other secondary endpoints were also included. No dose response studies were conducted in case of Flexavi, that is biosimilar. Efficacy study done only on rheumatoid arthritis with similar primary endpoints, that is American College of Rheumatology, 20%, that's ACR 20% response difference within plus or minus 50% margin in 95% confidence interval. Sample size is 584. That shows that ACR response at the same at week 30 was 64.1% and SB2 versus 66% in Flexavi. Secondary endpoints were also just measured or assessed on 54 weeks. When it comes to the conclusion, those response studies were not carried out by the biosimilar because it was already established by the originator. The clinical studies were done in comparison to the originator with similar primary and secondary endpoints. So the studies provided evidence of equivalence based on ACR response, that is American College of Rheumatology 20%. That's comparable. When we come to the reported adverse event effects, upper respiratory infections, headache, nausea, sinusitis, tuberculosis, rash, and cough were reported in case of Remicade, and also similarly the same reports of adverse effects were presented on Flexavi, that is biosimilar. Upper respiratory infection, headache, nausea, sinusitis, tuberculosis, rash, and cough were similarly reported or recorded adverse events. So, in conclusion, to the safety uptake, the safety profile and adverse effects were also comparable in the summary of product characteristics or SMPCs. So, when we come to the next one, just among the questions which are given for us to discuss, the next one is a reflection on how we would handle the licensing of that of biosimilar in all indications that were licensed for originator product, that is Remicade. To reflect, I think we choose to go back, to go back to the mechanism of action of the products, how they just used or treat the diseases. The mechanism of action, inhibition of tumor regulatory factor alpha, which is responsible for inflammatory responses of all the approved indications. The originator also carried out clinical trials on all the indications to prove efficacy for each of the approved indications. That is, we have seen just this reflection of handling of licensing comes from the existing data which are published on both comparative studies of that of biosimilar and originator products. For the biosimilar, also similarly, clinical trial required out for rheumatoid arthritis, which is the results were comparable to that of originator. Only, as we earlier mentioned, on the indication that was used for biosimilar, but is the only rheumatoid arthritis. Then extrapolation, when extrapolate or extrapolation is inferred on all other indications based on the similarity of the mechanism of actions across all the diseases. Because both the Flexabi and also Remicade are just acts with their mechanism of action are similar. Hence, the clinical trial study presented for biosimilar was or is acceptable. We come to the last part of our discussion, that is just to present a summary as to whether we would grant positive opinion for our agency or not, based on the information available to us. Just biosimilar products, Flexabi has been assessed for indicators of biosimilarity. Depending on that assessment, as per the available evidence related to pharmacokinetic, safety, and clinical efficacy data, Flexabi is similar to Remicade. So our concern is not on the differences on those similarities that are, even though the similarities or comparability is presented or that is confirmed by the clinical studies starting from pharmacokinetics to that of clinical safety and efficacy studies. But our agency needs bridging studies as ICH E5 guideline that requires bridging studies to cover differences in population before positive opinion can be made. That is what we decide to our agency based on the available data, and also what is just as a gap to be filled. That is bridging study might be required. So these are our just references to use that are also indicated on our case to refer those reports, summary reports that are posted on the IMA website. By this, I end my presentation. Thank you for your attention. That is the end of my presentation.